he studied other things besides that. And he's an expert in cellular physiology and molecular structure of these proteins. And then in recent years, looking at their contributions to behavior, including autism. And today he's going to tell us about flies, I think, and how you take mutations that are related to autism and learn some interesting things by studying them in an invertebrate. And probably also you'll talk a little bit about mice too, I suspect. Welcome.
Thank you so much, but I cannot share the screen.
Pramila, can you make it for him to share?
Oh, you can try again now. Be able to share.
Let me see.
Looks good.
Do you see the PowerPoint presentation, or let me see.
Yes.
Yes.
Do you see the comments, or you see just the PowerPoint presentation?
Just the presentation.
Okay. All right. So, well, thank you so much to the Brain Foundation and Randy for the kind invitation. And our group has a really strong interest in understanding how disruption of neurocircuitry or molecular mechanism can lead to neuropsychiatric disorder. In particular, we are interested in understanding the role of dopamine dysfunction as a complication or participating as a mechanism in the etiology of amphetamine abuse and for the purpose of this talk in autism spectrum disorder. So, for example, we are interested in understanding how amphetamine hijack particularly neurocircuitry in the brain in order to cause dopamine dysfunction and amphetamine abuse. We're also interested in understanding how single nucleotide polymorphism and de novo mutation in the dopamine transporter gene can disrupt dopamine transporter function, causing impairment in dopamine neurotransmission that we phenotype with specific behavior. And we use an array of tools to study these genetic variants, from structural biology to high-speed camera to look at the behavior of these flies. So dopamine dysregulation has been implicated in a number of neuropsychiatric disorders, ADHD, schizophrenia, neurological disorders such as Parkinson's disease, and lately also to autism spectrum disorder. In fact, some of the mutants that we analyzed in the past in collaboration with Randy Blakely has been found both in patients with attention deficit hyperactivity disorder and also in patients with ASD. So why ASD and dopamine dysfunction? There are a lot of traits that relates to autism spectrum disorder that points to dopamine dysfunction, from repetitive behavior, lack of reward for social interaction, hyperactivity, anxiety, and so on. So in the scientific community in the last few years, starting to publish paper looking at dopamine transporter dysfunction and his association with autism spectrum disorder. There are not many papers that have done that, but like I said, in the last few years, particularly our group, in collaboration with Randy too, has published a few papers regarding this subject. So just for giggles, there are two main dopaminergic projections. One is from the nigra to the striatum, and the other is from the ventrotegmental area to the nucleus accumbens and prefrontal cortex. What is important for this presentation is that the dopamine transporter is the main protein clearing the extracellular dopamine level once dopamine has been released. And really, it's been a renaissance for the dopamine transporter lately, because we discovered a lot of very interesting mutations related to neurological disorders such as Parkinson's disease, of infantile early dystonia, and neuropsychiatric disorders such as bipolar disorder, ADHD, and ASD, that give us really the opportunity to apply all the knowledge that we have acquired in the last 10 years about the dopamine transporter to understand really how this mutation disrupt transporter function and leads to specific behavior. So this is the topology of the dopamine transporter, is 12 transmembrane domain, and up to now, 5 mutants has been characterized in the dopamine transporter that been associated with autism. They're all in critical structural domain of the transporter. One is the N-arginine for a tryptophan at position 51 in the N terminus of the transporter. That is an important structural domain regulating transporter function. There is a deletion, an in-frame deletion of an asparagine at position 336 on the third intracellular loop. This is another mutation that is very, very important because the third intracellular loop is one of the loop regulating the intracellular gate of the transporter. The transporter have two gates, one extracellular, one intracellular, that are controlling and regulating the movement of dopamine across the plasma membrane. There is a alanine to a valine on the top of transmembrane 12. This was a mutation that was first discovered in a bipolar patient. Later on, a Vanderbilt and described by Randy and me for attention deficit hyperactivity disorder. And again, with the Vanderbilt patient in autism, again, characterized by Randy and myself. And finally, the first de novo mutation on the top of transmembrane 7, it is a threonine to a methionine. So when we look at the dopamine transporter, the physiological function of this protein is, how I said, is to clear extracellular dopamine for the extracellular space and move it inside. And typically, all this variation in pair transporter function, that is the one that we analyze, by two different modalities. The first modality is simply that the transporter goes on the plasma membrane, but is not capable to reuptake dopamine anymore, right? And so there is an impaired dopamine uptake. And the second modality that is much more interesting for me, because I'm a biophysicist by training, is that now the transporter runs backwards, right? For reason that can be signaling pathway or structurally, now the transporter binds dopamine from the inside, the neuron, it pumps dopamine outside, increasing it therefore extracellular dopamine level, and is what we call anomalous dopamine efflux. And this has been noticed for the first time in the A559V mutation, and later on in the T356M mutation associated with autism. So the proband is a male, has been classified as with autism spectrum disorder at age 5. And one of the important things about this threonine in position 356, it is highly conserved across phyla, from human to C. elegans. And the reason why it's so conserved, because he lies within the permeation pathway of dopamine and the co-transported ions, in this case, sodium. So you can imagine that any spatial constraint that the mutation threonine to a methionine can produce can impair, of course, the function of the transporter. And so like I said, we use a lot of structural tool from molecular dynamic simulation to structural biology, and also electron paramagnetic resonance spectroscopy to measure really how these mutations are impairing dopamine transporter function by looking on a structural perspective. So in collaboration with Samu Charab at Vanderbilt University, we do EPR, so electron paramagnetic resonance spectroscopy. So we engineer cysteine that are spin labeled, and EPR allow you to monitor the distance of these cysteines that we use it to look how the intracellular gate or the extracellular gate of the homologue, the bacterial homologue of the dopamine transporter is. And we look how these mutations are affecting the movement of the intracellular gate or the extracellular gate of this protein. And so without going into much details, what we saw that the intracellular gate was not really affected by the mutation, but we were looking at the spin label that we position on the extracellular gate of the transporter in the presence of sodium, the co-transported ions, and the substrate, in this case leucine, right? In the wild type, there was a rearrangement of the extracellular gate that was going in an occluded pore, right? So the transporter was trying now to move dopamine from the outside to the inside by closing the gate. But when we look at what was happening on the mutant, we saw a higher probability of these two cysteine, the two cysteine label, to be at higher distances, meaning that now the gate was opening to the outside. So when it was binding the substrate, moving the substrate outside the cell. So this was really the first structural fingerprint of what anomalous dopamine efflux was, meaning that the transporter, when it binds the substrate, spewed the substrate outside. So we had to choose a model now, an animal model, to start to screen all this mutation. And of course, mice cost a lot. And we were running on Fume on these projects, right? So we choose Drosophila to express the human dopamine transporter or the mutated dopamine transporter in the right neurocircuitry, meaning the dopaminergic system. So we use the US GAL4 binary system now to express, like I said, the human DET or the mutated human DET in dopaminergic neurons of flies that are lacking the Drosophila dopamine transport. And so the first thing that we've done, right, is to look at the fly brain that you see over here. There are a lot of dopaminergic projections. You see them in red, and to start to figure it out how to study anomalous dopamine efflux. Again, the transporter running backwards. Before the work was done only in cells, and now we wanted to move, of course, to an animal model. So what we use is amperometry, is a technique that takes advantage of a carbon fiber that we position in any dopaminergic node of the fly brain. And this carbon fiber measures the level of dopamine by oxidation, right? It is represented as a positive current. So you can expect that if you have an anomalous dopamine efflux, the baseline recorded by this electrode will be higher than in the wild type. You will also expect that if you block the transporter, either with methylphenidate or other blocker like cocaine, this baseline goes down because you're blocking the constitutive dopamine efflux of dopamine, and this is what we notice over here, right? So you can see that you're already cocaine and the baseline goes down because you're blocking the constitutive dopamine efflux of dopamine. And we've done several studies characterizing structurally and the signaling pathway regulating the anomalous dopamine efflux, including we saw for the FFNV mutation, there was phosphorylation of the N-terminus of the dopamine transporter. So what does it mean, this behavior? The first thing that we did is to convince ourselves that flies was a good model to study neuropsychiatric disorder. And we asked the help of a very famous scientist, Charles Darwin, and in one of his books, The Expression of the Emotion in Men and Animals, said that even insects express anger, terror, jealousy, and love by their stridulation. And so we thought that we were on the right track. And so the first thing that we looked, it was how the anomalous dopamine efflux was influencing locomotion, because a lot of patients with ASD are also hyperactive, right? And so I want to show you a movie. This is video tracking of flies expressing either the human DET or the human DET that now has been mutated. And you can see that the flies have the human DET causing anomalous dopamine efflux. They are moving much faster. They're crossing the center of the cage much more. And they're grooming much more than the flies. They're just expressing the human DET. And this is the path that the flies that are expressing the human DET with the mutation are performing during this time. You can also do beam crossing to look at the circadian locomotor activity. So these are the wild types, so the human DET over at the turn on of the lights and the turn off of the lights over 26 hours. And you can see clearly that the human DET expressed in the T356M is completely hyperactive, almost similarly to flies that are not expressed in the transporter at all, and therefore have a really high level of extracellular dopamine level. And you can quantify this over 24 hours to see clearly that the mutation causes hyperactivity in these flies. So another characteristic in autism that a lot of young people with autism spectrum disorder have a really high level of anxiety, with fear to be one of the most common subtypes of anxiety in autism. And so the question is really how to measure fears in flies, right? Animals, I'm a really outdoor person. An animal, I know that they fear under predatory pressure. And they fear to camouflage, but they fear also because they are scared, right? And the question was how to scare flies, right? And one time I was in my cabin where I pick a lot of mushrooms, right? And I saw one of these guys is a predatory wasp close to a nest of flies. They emit a sound like this. I don't know if you can hear it. And so the flies are scared. I'm sorry to turn off the sound because it was making me deaf. And the flies are scared simply because the predatory wasp is eating their larvae. And so what we did, we recreate the sound of the predatory wasp in the lab. We use a high-speed camera. This camera can record 50,000 frames per second. This is a video that we recorded at 3,000 frames per second. And if you don't see here the sound of the predatory wasp, there is a red highlight of the cage that will tell you that the sound of the predatory wasp is coming on. And we're measuring freezing behavior in these flies, right? So you can see the flies move around, the video is being slowed down, the sound come on, they freeze, and they escape, right? And this is a natural behavior. Usually animals freeze, and then they look in the environment, they escape. And so this is the mutant instead. And you can see that the freezing time of this of the mutant is much longer than the wild type. You can even notice it by eye. So the mutant moves around. It really doesn't move anymore, right? So you can quantify this average velocity of the fly every millisecond, right? And this is the wild type in black. The animal moves around, the sound of the predatory wasp comes on, the animal freezes, and then is escaping. Instead, if you do the same type of experiment with the mutant, the animal moves around, the sound of the predatory wasp comes on, the animal freezes, and it keeps staying in a freezing state. Finally, we wanted to look at social interaction in autism that are impaired, looking again at what anomalous dopamine efflux does to this social interaction. So we have to find ways to measure social interaction again in flies, and there are several ways. I thought that this would be a cool way to look. You know, if you look at the school of fish or flocks of flies or birds, they maintain social distances. They maintain social distances because they try to survive in the environment, particularly when a predator is around. This is a school of fish that's being predated by a duck in the back. You can see it over here. And one thing that you notice is that the school of fish compress and expand depending on the distance of the duck, right? And so this is a behavior that is being modeled to say that when the animal, the predator approach the flock, the school of fish, the school of fish will compress and then it will immediately expand because they try to escape altogether away from the predator. So we thought that we could do the same using the flies, meaning that we can measure the social distances of the fly, the cumulative social distances over time before and after the sound of the predatory wasp. And so this is what we saw. So this is the wild type, so expressing the human death. And it's at the on of the sound of the predatory wasp, they compress and then they expand. And if you look instead at the mutant, first of all, they start from a compressed state, right? But at the on of the sound of the predatory wasp, they expand and then they compress. So they do the opposite of what the wild type are doing. And so what we are interested in is not only about the human dopamine transporter, but also other proteins, right, that participate in the signaling pathway that are regulating dopamine transporter function. As we talk, we're working on syntaxin 1A. This is a protein that regulates the ability of the transporter to reuptake dopamine. Today, I show data that parallel a mutation, the first novel mutation in the dopamine transporter, that cause anomalous dopamine efflux. We characterize this mutation structurally, so we fingerprint what anomalous dopamine efflux occurs for this mutation, that is the opening of the extracellular gate, allowing the dopamine to be released outside. We also measure dopamine efflux, anomalous dopamine efflux straight in the brain of flies, and link this to impairment in specific behavior of the fly, including increased locomotion, increased fear response, impair social distances. Of course, the question remains, what is happening to more complex behavior that cannot be studied in flies? And so we have done a knock-in mouse, right? And we show in the knock-in mouse that we have impaired dopamine clearance, decreased tissue dopamine content, increased locomotion, impaired social behavior, and loss of social dominance. And you can see all the data published in a recent article in Journal of Clinical Investigation. I do have to say thank you to a lot of people, particularly Gabriella Di Carlo, Peter Hamilton, Aparna Shekhar, Jenny Aguilar, and Christine Saunders, Andrea Belovich, and outside the lab to Greta Galli, my daughter, that did some of this experiment, Kevin Harrigan and Einar Mattis. Einar Mattis is an expert in genetics of flies. Mark Wallace Urrigeter, the geneticist James Sutcliffe, and finally for the EPR to Assam Mucharab and Shruti Sharman, and for you all for listening to me. Thank you so much again.
Thanks, Aurelio. It's just amazing to see these conservation of these behaviors. Obviously, you know, a fly is not going to be a mouse, is not going to be a human, but we can trace these different systems, as you showed, you know, nicely with the fish and the flies, and the way in which we have protective behaviors ourselves together. It's just amazing. I have a couple of questions just of my own to remind me. And, you know, I'm sorry, I should remember all of this, but in the fly model that you introduced us to, was that the human mutation in the fly background, or was it in the human background?
It was, so we express the human dopamine transporter directly in flies in dopaminergic neuron. So these dopaminergic neurons are lacking the Drosophila dopamine transporter. So quote unquote, the flies get humanized with the human dopamine transporter.
I see. And do you put the wild type in parallel as your control?
Yes. So
okay.
Yes, and we backcross this fly quite a bit. So.
And does the animal with the human dopamine transporter look, and just the wild type, does he look just, or does he behave just like a wild type fly with a fly transporter?
You know, we have done this for locomotion, and they do, right? You know, the high-speed camera experiment are really complicated, so we don't have a parallel with the Drosophila dopamine transporter, but we always compare it to the one expressed in the human dopamine transporter. In terms of uptake and efflux, they are the same. So the one expressed in the human dopamine transporter are really similar in terms of clearance and response to psychostimulants, for example, to amphetamine, to what we have seen with the flies expressed in the human dopamine transporter.
So maybe with time, because these other domains that are probably divergent have regulatory elements in them, and you might find some really interesting things where the transporter is regulated, maybe not appropriately because of the sequence divergence.
Yeah, we are working heavily now on the microbiome to look how it's influencing transporter activity. Right. So.
Right. Yes, Dr. Galli has quite a nice program going on microbiome, and with his dopamine background as well. If anybody's interested in asking about that either now or later, I'm sure he'd be interested in hearing your questions. I have one more question about flies and how you use them. So a lot of people will turn to Drosophila because they can screen for mutants or screen efficiently, you know, genetic stocks that have mutations in a whole bunch of things. Have you explored yet any of these genetic variants that might, you know, if you put them together with I think that your T356 mutation, this de novo mutation from individuals with autism, is a very powerful reminder. I mean, it's powerful evidence that the dopamine system can contribute to autism, whereas the rest are probably more risk factors and need other things to come together. So that was what sort of made me think a bit about some of these other mutations that might be interesting to put together. Have you put any together with yours?
No, but you know, the T356M is actually found now in another patient in Cincinnati, and we're in contact with the doctor. They just swab the saliva and send it to us to look at the microbiome, and also in a new patient in Europe. So it's becoming a very interesting mutation to follow up in all these kind of things. In the mouse, for example, the one that I show in the knocking mouse has a difference in the fusobacteria in the gut, right? It has been linked to dopamine dysfunction in autism, and we measure the microbiome in this mouse, and now we're trying to compare this with the patient.
Dr. Blakely, there is a question in the Q&A.
Okay, let's see. Can I find it here?
Can
Okay, what do we know about neuromotor issues in the mutants? How do we go from DAT to behavior response? Aurelio, you showed us hyperactivity. You also showed us repetitive grooming behavior. Any other motor responses that might interest this audience in relation to?
So in the knocking mouse, they are performing much better on the rotor rod. That is another motor response that I remember now on the top of my mind. And the mouse is hyperactive too. I try to remember other things. In the social dominance test, they lose, like you show for your mutant and the serotonin transporter. That is about it that I remember that we've done. And the mouse is available to everybody. So whoever is interested in using this mouse, just email me, and I'm more than happy to send it to you.
So maybe, Ashok, if you had a more refined question about neuromotor issues, were you thinking about something in particular? But Ashok has another question about that microbiome. He wonders about specific metabolites that might be secreted that could be responsible, I think, for some of the behavioral changes that you're seeing.
What other metabolites do you have in mind?
Are there bacterial produced metabolites that might be affecting brain function?
Okay, so we didn't look at that. So the fusobacteria is producing butyrate, and we have a model in actually amphetamine abuse, because a lot of methamphetamine abuser has increased fusobacteria in the mouth. That is why they have gingivitis. And so when I was reading and finishing up this paper, hopefully soon, on butyrate and amphetamine and methamphetamine abuser, I discovered that in autism, it looks like this kid has low level of fusobacteria. So we got a knocking mouse, and this was the first thing that we've done, is to look at the fusobacteria in this knocking mouse, and they are low. We didn't measure butyrate because it's actually quite difficult to measure. Right?
Well, that's going to be interesting to follow up. One of the things on the serotonin side, serotonin obviously in the gut affects the microbiome as well. And we've been trying to get our mutation in a cell-specific knock-in, which is a little harder to do than the transgene or just a standard knock-in to do it conditionally. But I have wondered how much of the behavioral phenotype that we have is due to what's happening in the gut being transmitted to the brain, just like the immune function, right?
Yeah, in flies, butyrate influence a lot of this behavior. It's influencing courting behavior, is influencing locomotion. So we feed the butyrate directed to the fly, and we modify the behavior of the fly. Now, we didn't do it in the mouse.
It's an interesting story about amphetamine, and I understand why you would do that, given DAD as the site of the mutation. Let's see. I think that's the end of the questions that I see. I'll thank you again, Aurelio, for
thank you so much.
A very interesting talk and providing this insight, this evolutionary insight into how we study autism mutations. And now move to our next speaker. So our next speaker is Harumi